This protocol facilitates the successful structure determination of the IKK-binding domain of NEMO in its unbound form and details of its production and crystallization. The protocol takes advantage of the stabilization of the native conformation of the IKK-binding domain fragment of NEMO through coil-coil adapters, which facilitate crystallization and structure determination.
The structural biology of NEMO as a target provides an important advantage in the development of NEMO inhibitors for the treatment of inflammatory and autoimmune diseases and cancer.
This protocol may be extended to the structure determination of NEMO complexes with peptide or small molecule inhibitors for drug discovery or optimization. The protein refolding and concentration during the protein production stage are fundamental to obtaining a pure NEMO dimer, limiting aggregation and ensuring solubility under crystallization conditions.
Demonstrating the procedures will be Tamar and Amy, graduate students in our laboratory.
Begin by adding 20 milliliters of terrific broth solution and 20 microliters of a 100 milligram per milliliter stock solution of ampicillin to a 125 milliliter Erlenmeyer flask, followed by a few microliters of cell glycerol stock from minus 80 degrees Celsius storage of BL21DE3 competent cells transformed with vector. After shaking the starter culture overnight at 37 degrees Celsius and 220 rotations per minute, dilute the cells to an ODC.